In the next 3 minutes, I'm going to talk to you about our recent paper published in the Journal of Cell Science entitled Transcriptional Adaptation of the Deletion of Cdc42 in Primary T Cells. Gene deletion is a common paradigm used by cell biologists when investigating protein function. This works by comparing a wild-type cell with normal gene function and a knockout cell without that gene and seeing the difference in function between cells. The conclusion being that the targeted gene is responsible for the missing function in the knockout cells. We set out to leverage this paradigm to ask the question, what does the protein Cdc42 do in cytotoxic T lymphocytes? Now, Cdc42 is known as a master regulator of cell polarity. This small protein is responsible for controlling the cytoskeleton in various ways. Cytotoxic T lymphocytes are a type of T cell that are responsible for killing cancerous or virally infected cells. We suspected that Cdc42 was doing something important in T cells because when the gene for it is mutated in humans, it causes immunodeficiency. So when we knocked out Cdc42 in T cells using CRISPR-Cas9, we were surprised to find that the T cells actually had improved function, not worse function, across killing, degranulation, and T cell receptor signaling. However, when using a chemical inhibitor of Cdc42, which we showed was highly specific, we found that the cells were worse at performing their job, which was more in line with what we had originally hypothesized. A key insight to explain this discrepancy between the inhibitor experiments and the gene deletion experiments came when we did a time course of killing assays daily after nucleoaffecting the cells with the CRISPR machinery. We found that initially the Cdc42 knockout cells were actually worse at their function, like when we inhibited them, but gradually they regained their function over time. Comparative transcriptomics and proteomics showed that genes were upregulated in the knockout cells, which improved their function, a phenomenon known as transcriptional adaptation. One of the ways in which this worked was by upregulating the activity of related proteins to Cdc42. Transcriptional adaptation only occurred when we used CRISPR-Cas9 to chop the middle of the Cdc42 gene, and didn't occur when we targeted the promoter region of Cdc42, which was consistent with what others had shown in different systems. Overall, our data highlight an important limitation for CRISPR-Cas9 based investigations, while also showcasing an interesting way in which T cell function can be enhanced, for example in cancer immunotherapies. If you'd like to dig deeper, feel free to find the full open access research article on the Journal of Cell Science website. Thanks.